My name is Bertie Gottgens. I'm the director of the Wellcome at MRC Cambridge Stem Cell Institute, and I also run a research group in the institute where we study the biology of blood stem cells, how blood stem cells develop in our bodies and how they maintain the blood system, and also how, when things go wrong in blood stem cells, how this could cause leukemia.
Great, thank you. And can you tell us what work you've been doing on COVID during the pandemic?
On COVID, it of course was a major shock to all of us having to shut down the labs. But then I got to understand very quickly that the immunologists in Cambridge had started an amazing effort to collect blood samples from the COVID patients that were admitted to the hospital in Cambridge. And so I then contacted them and linked up, and we set up an entirely new project in collaboration with the immunologists in Cambridge, researchers from the University of Newcastle, and also the Wellcome Sanger Institute just outside Cambridge, where we applied a new way of analyzing those blood samples that we had developed for our work on normal blood stem cells. And this is a very high profile, high throughput way of profiling the molecular makeup of individual cells. And in the end, we applied this method to study more than 100 patient samples and more than 700,000 individual single cells. And all of this information was then data crunched to extract a lot of take-home messages out of this to say, well, what can we see in the blood that's different between patients that have very severe COVID or mild COVID, etc. And this then laid the foundation for a lot of subsequent work where we still contributed to it, where, for example, the question was asked, what's different about the blood in young children that have COVID compared to adults that have COVID? So it was an amazing sort of collaborative effort, and also meant that the lab actually wasn't shut for very long, because after 2 weeks we were back in, now applying our skills to learn something about COVID.
And is that work still ongoing?
So at the moment, this work is not ongoing anymore. The data sets are all published and they are being used by other groups to interpret whatever they see when they're looking at COVID samples. But just to illustrate the sort of cycle of biomedical research, from performing the research to actually seeing it published in a journal, does take a while. So the study on the young children with COVID was only published around Christmas.
And tell us about one of your other recent science highlights away from the COVID work.
So one line of work that I'm really interested in, is currently excited about, is currently ongoing in the lab, is that we're trying to add a time component to our understanding of how blood cells develop. So what we know about blood cells is that it's this hierarchical system where blood stem cells, they continuously replenish the blood system. So an average human being makes around 3 million new blood cells every second. They also lose around 3 million blood cells every second. So this replenishing is driven by blood stem cells that can make all the different types of white blood cells, red blood cells, the platelets that you need for clotting. What we don't know is, under normal circumstances, how long does it take for a blood stem cell to divide and divide and divide to make a number of red blood cells. So we're currently trying to learn this time component. And I think it's very important because in diseases, it's the time that is perturbed a lot of times.
So Betty, how has life as a scientist changed over the past 2 years for you and others in the Institute?
I think the biggest change to your everyday life as a scientist has been to adapt to home working, hybrid working, and then to learn to communicate with each other much better while you are working under this new normal. So that's it offers challenges, it also offers opportunities. For me personally, my life as a scientist has also changed because I've taken on new responsibilities by taking on the role of director of the Stem Cell Institute at the end of last year. So there are new and exciting challenges that come with any leadership role of this sort of type.
Great. And what would you say the best lesson you've learned is from the past 2 years that you can take forward in your research?
So I would say the best lesson is that we were able to move very fast with the COVID research because we had managed to assemble a collaboration across 4 different institutes: the Immunologists and Stem Cells in Cambridge, the Wellcome Sanger Institute, and the University of Newcastle, all working towards a common purpose and pooling their resources, interdisciplinary expertise. But yes, I think that's a big lesson how this has allowed us to achieve something that none of the individual entities alone would have been able to achieve, and to do it really rapidly.
Great. And have you seen increased interest or scrutiny of biomedical science like yours since the start of the pandemic?
So what I've seen, and obviously to some extent also done myself, is that the whole world seems to be interested in medical things, biomedical things, that I can now talk to somebody in the street and say, oh, I've had a PCR test, and they know they have some idea what I'm talking about, whereas 3 years ago, 90% of the population would never have heard the word PCR. So this is clearly a change. And if something affects you personally, your personal life, then no wonder that people are interested in this. There are problems and real benefits with this. I think the problems are that in our sort of society that's driven by online communities, there can be misinformation that's being pushed, or people having agendas and they're using some sort of scientific language to push the agenda. And at that point, I think it's really important that people remain trustful in the actual scientists who can then say this is above board, whereas something else is really a misinterpretation of the results.
And leave us with a hope for the future based on the experience you've had over the past 2 years. What do you hope will change, or that you'll be able to keep up?
It's my impression that the general public at the moment cares about medical research and understands how it is important in how it can make a difference in our everyday lives. This is important for us as biomedical researchers, to have the public on our side. And what my hope is that through excellent public engagement and communication activities, that we can just maintain this trust.